Distinguished guests, fellow students, present and past, Professor Chitnis or Dr. Chitnis, we welcome you to the second lecture of Faculty of Architectural Alumni Association, which is an organization which is being set up. School of Architecture is completing 50 years in 2012. And we are planning to celebrate that in a big way. By now, about 1,000 to 1,200 students must have passed out of this school. We all want to be associated, contribute, and get connected with the school which has given us a lot. It is this bond and this desire we are trying to articulate in form of Faculty of Architectural Alumni Association. And as part of this activity of this organization, one of the things which we would like to do is to bring several distinguished people to come and talk to us, share, and continue the process of learning. As part of this, we have invited Chetan Chitnis to give us a lecture today. And we hope to continue this process year after year so that we can get connected and have a long-term relationship with the educational process in this school. I invite Bimal to take over.
So welcome everybody. It's an honor and privilege to introduce Chetan Chitnis. Chetan is an Ahmedabad boy and a childhood friend of mine. We first met when he was in the 5th standard. I was in the 5th standard. Before that he was in GLS, I was in Xavier's. And he came one day with his friends from Xavier's to swim in the pond there. And he met Father Zavala. He liked the place and he asked Father, in those days things were easy. He asked Father Zavala, I want to come here from tomorrow. Father said, okay, from tomorrow you come to Xavier's. So that's how he joined Xavier's. His official biography starts with his college education, but I think his journey in science started much earlier. Both of his parents are scientists. His father is a physicist, and he was at that time at the Space Application Center. Eventually, he headed the Space Application Center in Ahmedabad. They were all Vikram Sarabhai's people. Chetan's mother studied biology, and she was at the cancer hospital, and she worked all her life at the cancer hospital in Ahmedabad. So both his parents were scientists. So he had a background in science, so to say. Journey in science, I think, began already in high school. One of the finest things that happened to our batch was that we were the batch that was the first 10+2 system batch. And so we were shifting from the 11-year SSC to the 10+2 HSC system. As a consequence, right through 8th, 9th, 10th, 11th, and 12th standard, we had no textbooks. So that required that a lot of learning had to be done on our own. And I think that was very important sort of thing that led to some of the learning at that time. The most important institution at that time was the Community Science Center. Because we had no textbooks, we had to use the library in Community Science Center. And the laboratories there were also great places, a great place to do a lot of work. So Chetan's journey in science begins next door from Community Science Center. We had a brilliant science teacher in the 8th standard, 8th, 9th, 10th, 11th, 12th, in Father Morondo, who taught biology. And that was, I think, the first time that you heard of genetics. At that time, Community Science Center was a really vibrant place. There was no internet. There was not such things like the internet. And you couldn't get many things, but there were many lectures there, and it was a vibrant place to learn. The laboratories were also fun to work in, and in the school they encouraged people to go to the laboratory and do lots of things on their own. Chetan finished his HSc and headed off to IIT Bombay, where he got an MSc in physics in 1983. Actually, that's while he was at the IIT, he used to keep coming back home, and he used to hang out at the Sept canteen very much. So he knows the Sept chai, and he knows the taste of the Sept chai quite well. After finishing his MSc in physics in 1983, he headed off to Rice University, where he did an MA in physics, studied some more physics up to 1986. Then he joined the biophysics program and did a PhD in biophysics from the University of California at Berkeley, which he completed in 1990. Having finished that, he headed off for a postdoctoral fellowship in the Laboratory of Parasitic Diseases at the National Institute of Health in Washington, where he started working on malaria for the first time. Chetan returned to India in 1996, where he established a vibrant research group at the International Center for Genetic Engineering and Biotechnology in New Delhi. There, ever since he returned in 1996, he has been working on developing a malaria vaccine. And the vaccine that they have developed will soon enter clinical trials. Chetan's work is funded by the Department of Biotechnology, Wellcome Trust, the Gates Foundation, Howard Hughes Medical Institute, and the European Commission. Chetan has published over 70 peer-reviewed papers in international journals. He's a member of the editorial board of several journals. He has served as a member of prominent international scientific advisory committees, such as the Disease Reference Group on Malaria of the World Health Organization. He has been invited to present his research at several international conferences and academic institutions, and served as chair for the Gordon Research Conference on Malaria in 2009. He is recipient of fellowships from the Wellcome Trust, Howard Hughes Medical Institute, USA, and Department of Biotechnology. Chetan received the Young Investigator Award from American Society of Microbiology in 1995, the M.O.T. Iyengar Prize from ICMR in 2003, and the Shanti Swarup Bhatnagar Prize in Medical Science from CSIR in 2005. Last year, he received the Infosys Prize in Biological Sciences, which is a big award established by Ms. Narayan Murthy. The latest issue of India Today, if you see it, has Salman Khan without a shirt on the cover. Inside, they have an article. It's the 35th anniversary issue. Inside, they have a section on scientific heroes. One of them is Chetan Chitnis. So here we have Chetan.
Hope that the work we do eventually has an impact and a positive impact on society, and we really need to engage with society. And society needs to engage with science, because I will try to show you throughout this talk on biotechnology how many of the things that are happening. So, exciting things happening in science, and as members of the society, we need to decide decisions and make opinions about what should be, because a lot of this will impact you as members. So start at the very beginning and start with the question, what do biologists think about? What kind of questions do they ask? Well, here are some of the questions they asked. They asked pretty big questions. They'd like to know, can we have these lights off? I think these two. Okay, thanks. So the kinds of questions they're interested in asking is, what is the origin of life? Where do we human beings come from? Where are we going? How do living organisms work? And from what we learn by understanding these basic questions, what can we do to solve problems that we face? So how can we use this knowledge to solve problems that mankind faces? So starting at the very beginning, the most accepted theory for the origin of the universe, not just we'll get to origin of humans, is the Big Bang theory, which says that all the matter that there is in the universe started off, was all coalesced at one point, and has been expanding now for the last, I don't think this is working, for the last 15 or so billion years. And during this process, the universe has been cooling, the universe has been expanding, and a number of galaxies, stars, etc. have formed. And this time frame of about 14 billion years is based on measurements made on the radiation in the universe, on the matter in the universe, etc. So that's how long the universe has been around. Now, you would imagine that the process like this would be chaotic. This expansion looks like, and start forming. Now, or speak new. Origin. See beautiful again, because these blowing blowing is something close, and then if a predator comes, he'll. So patterns emerge. Have to do the origin of these patterns. But how can this explain? Well, let's think about pattern conditions. The form it was predicted there would be ammonia, there would be hydrogen, there won't be any oxygen very early on, and then there were volcanoes, so there was fire, there was lightning, and one young student, Stanley Miller, in the early 1950s, in 1953, thought of doing an experiment. And the experiment he thought of doing was, if I create the primordial atmosphere in a bottle, given the gases there might be, given the lightning, if I simulate all that, what kind of molecules might emerge? And this is the experiment he did. So he built this equipment. The equipment is actually what you see in the previous slide. So this is his equipment that he built as a graduate student. And this is the experiment as a schematic. So he had this bottle with gas he filled with.
go for and our question pulls up radial atmosphere. At the end of a week, he saw this brown detected acid. Now, what are amino acids? Building blocks of all proteins. So, basically, running the cell, protein to cells, they run to it of amino acids. So, there are strings of amino acids from the nitrogen and suddenly 11 and finally was a very all the from much more sensitive techniques. Take a look again at had been able to create 50 years ago. He actually detected not just 5 but all 20 created. Building blocks of all organisms 20 had now this actually started off who had the same experiment showed that you can look at other molecules on the basis of membrane and nucleotides get to what those are. There is an amino acid. The only chemical groups that's that acid you get these the structure for what it does it's together to form proteins. Things phospholipids. There's this long of the acid is charged. If you add it to water, so this groups are inside and this charged group. The reason this is important is these bubbles to or bubbles very important for the phosphorus. Which then of nucleic acids are the A, T, C, and first this looks like hydrogen by from Cambridge 1950s, and this is the first structure they put together, and they actually predicted that this would be a replicating molecule. And the reason it can replicate is because you know if on one strand you have an A, on the next strand you have to have a T. If on one you have a G, the next strand opposite have a C. So when this molecule replicates, you can actually create two different molecules. And the reason this is important is once you have a replicating molecule, that brings into play natural selection. So if you have replicating molecules, nature will select for replicators that are more efficient, that are more stable, because they will win the race. So that's how a replicating molecule in evolution is extremely important. It was the next thing. The other important thing is that replication is not perfect. Replicate is a mistake, and that's what leads to diversity. Or were able to replicate molecules better would be the expected work. And this is actually based on 50 years of study. I'm going to show you a movie about how DNA replicates itself. Let's see, hope this works. So that's DNA. Missing.
Able to see how DNA is actually copied in living.
So these are proteins.
You are looking at amazing miniature biochemical machines, DNA double helix, and cranking out a copy of each strand.
Two copies made.
Enters the production line from bottom left. The whirling blue molecular machine is called helical. It spins DNA as fast as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right.
This is a strand. A copy has been made here.
Things are not so simple for the other strand. It must be copied backward. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
So this is happening in your cells. Every time a cell in your body divides, your DNA is being replicated. And it's an amazing machinery to copy it accurately because you have two strands. So from one strand, double helix, you get two double helices, and one will go off to one new cell and the other one to the other. And this is, it's animation, but actually this is based on sort of 50 years of work and a number of Nobel Prizes for identifying which molecule does what. You know, the helicase, which unwinds DNA. Then there's a DNA polymerase, which will come and add new nucleotide strands. So this complex machinery has evolved to replicate DNA. And also, again, I'll remind you, it's not perfect. It will make errors. You make an error in about 1 in a million bases. That's what leads to diversity. And that's what's happened with evolution and the diversity you find in all these organisms you find on Earth. Each one has succeeded in its own natural environment, in its niche, because it was successful at replicating DNA. And that's the diversity you see on Earth. Now, so to look back again, so evolution itself has different stages and has evolved. So there's been evolution of evolution. So first of all, you had the phenomenon of emergence. I first talked about it galactic scale, but then at the molecular level, you had the emergence of building blocks, the nucleotides, the amino acids, the phospholipids, and then creating the first next step. The next big step was the creation of a molecule that could replicate. Because as soon as you got replication, self-replication, you brought in natural selection. And natural selection, all that was a process of optimization to select best replicators, most stable replicators, fastest replicators. And that's what is evolution very, this is again very commonly understood as natural selection, the one that survives, the fittest who survive. Now this went on for many generations and you got this diversity on this planet. And the next big step on evolution was the appearance of humans and the evolution of brains. Because what brains brought, in human brains, was intelligence, the ability to reason, the ability to plan, and most importantly, the ability to build technology. Because what that allows you to do is allows you to grow food, allows you to build shelter, and that takes away natural selection as the optimization process for humans. Because you're no longer at the mercy of nature. You are, of course, at times of earthquakes and floods, etc., but on the whole, brains and intelligence and the ability to reason allows you to negate natural selection, and that has been the next big jump if you look at the rate of change. I mean, 100,000 years ago, it's estimated there were only about 100,000 humans, the first Homo sapiens. And look at what's happened in the last 100,000 years, for better or for worse. We are 5 billion, 6 billion people on Earth and have changed the face of the Earth. And the next big step, and I've put a question mark, but that's sort of going off into the realm of science fiction, is our ability to build technology which might actually do better in thinking with artificial intelligence. And what if you create artificial intelligence or computers that can replicate themselves? And you wonder what that will happen to evolution. But coming back into the realm of science, let's go back and now think, so the evolution of a replicating molecule of DNA was a very important step in evolution, followed by the appearance of humans and the evolution of brains. But let's go back and look at why is DNA so important? Well, the reason DNA is important is because DNA in any organism or any cell is what carries the program of life. Now, in computers, you write your program in the binary code. Think of it in the code of life is written with the genetic code with these 4 bases, A, T, C, G. And by what I mean is, the important thing, remember, is proteins. Proteins do all the work in cells. So how does DNA encode proteins? And that was figured out by, again, work of many, many scientists. But the DNA, they were triplets. So you have A, T, G, and 3 base pairs encode and tell you what protein to make. So in a gene, the sequence of DNA, the sequence of these base pairs tell you what string of amino acids to make. And how does that happen? Well, again, you can think of this in the analogy of computers. So DNA is like your hard drive, whereas you want to get to proteins, which is, DNA. In computers, you manipulate information. Here, you're manipulating matter. And to get from DNA to proteins, there is an intermediate step. And you can think of that as creating RAM, or random access memory. So from DNA, you first make RNA. And RNA, again, is made of bases. And again, they pair the same way. The only difference is instead of T, there's something called a U. So if you have an A there, you'll get a U here. If you have a G there, you'll get a C. So again, it's complementary to the DNA. And this step is referred to as transcription, going from DNA to RNA with the help of an enzyme of a protein called RNA polymerase. And from RNA, this RNA comes out in the cell and goes to a molecular machine called a ribosome, where there are these small adapter molecules, again RNA, which have only 3 bases. And again, they are on one hand attached to an amino acid. So for each amino acid, there is a transfer RNA, and they then base pair with this RNA. And the sequence again here will determine which sequence of amino acids you string together. And again, the details of this process are again pretty well worked out. And so let's first look at transcription. So DNA being copied to RNA.
What you're about to see is DNA's most extraordinary secret.
So that's your
How
DNA
a simple
with the
code
gene.
is turned into flesh and
Here
blood.
are some proteins.
It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands.
That's your RNA being made there.
The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
So now your RNA is made. This RNA will go out and find a ribosome, and it will get translated or turned into protein. And let's see how that happens. So there's your RNA.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time.
That's the protein molecule.
And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
So again, amazing small molecular machines in each of your cells producing proteins all the time. And what is the genetic code? So we mentioned that a triplet of bases encodes for one particular amino acid. Well, that was figured out by mainly these 3 scientists, including Hargobind Khurana at MIT, Marshall Nirenberg working at the NIH. And they figured out, so if this is the genetic code, sort of this is the key of life. If on the RNA you have a UUA, you get a leucine, which is one kind of amino acid. If you have a UAU, you have a tyrosine, another kind of amino acid. So depending on the string of amino acids, you'll put string together amino acids in the sequence that the DNA sort of encodes. And then you will notice you also need to know where to start a gene and where to stop it. So there are some codes for stop. So UAA means it's time to stop. And actually this was figured out by a scientist, again with roots in Ahmedabad, Anand Sarabhai, for his PhD work at Cambridge in the 60s. And these were the 3 who figured out mostly the genetic code and got a Nobel Prize in the mid-60s. Now proteins on their own, they're not just a string of amino acids, but based on the chemical properties and physical properties of that string, they actually fold. There are interactions, and folding of proteins is very important for them to actually become functional. So all our cells, all the stuff of life, sort of the the production of energy, the structure, etc. This is all encoded by proteins. And this is what the cell looks like. These are actually real cells stained with different markers. And these are the different organelles within the cells, the nucleus, the endoplasmic reticulum, and a lot of stuff goes on in the cell. And I don't have time to sort of explain all of it, but let's just watch another movie and see all the things that go on in cells. And I'd like to remind you, these are movies, these are animations, but they are scientifically extremely accurate. The structures of each of those proteins you saw for DNA replication and production of proteins is actually been determined. So there's the work of literally hundreds of scientists over the last 50 years. The structure of the ribosome, which makes proteins, the three people who got the Nobel Prize just a couple of years ago included, again, from Cambridge, Venki Ramakrishnan, who was instrumental in solving the three-dimensional structure of proteins and explaining how that process happens. So let's look at what all goes on in a cell. Lymphocytes rolling along in your blood vessel. These are two different cells interacting with each other. These are proteins on the two cells making contact. So cells communicate with each other. And this is a closer view of proteins on the membranes. And this is now looking inside the cell. And these are all proteins, all doing their jobs, their specific jobs in the cell. This is the cytoskeleton, the spectrin, gives structure to the cell. These are some more proteins called actin filaments, which again play a very important role in providing structure inside the cell. And these are all very dynamic. They sort of assemble and disassemble as needed as the cell grows and faces different environmental conditions. So for example, here, this will come and break off this actin filament. These are microtubules. Again, assembling, and microtubules are like highways inside the cell because you need to transport proteins from inside. Sometimes proteins have to be exported, and they're exported in these big vesicles. And here is a machine to move vesicles within the cell. Here is a mitochondria. Here is the nucleus. This is the RNA being made and snaking out. And here is a ribosome assembling and a protein being made. One protein being carried off into the mitochondria to do something it needs to do there. Another protein that's being made is actually, if you want to secrete a protein, they secrete it first into these organelles called the endoplasmic reticulum. And then little vesicles bud off and then they move towards the surface. There's another organelle in the cell called the Golgi. And fusion will take anything going to the surface, goes through the Golgi, and here are proteins being released from the cell. They come to the surface, and again, these will interact with this other cell. Again, are flexible based on conditions. They often need to move. And here's a cell. Now, if it senses an infection, there's a lymphocyte. It changes shape and sort of finds its way out of the blood vessel to go to the site of infection. So again, this is work of people trying to understand cell biology, biochemistry within the cell, how cells communicate to each other, and put together to show you what are all the things that happen that a cell does. And so if on any day if you're feeling lazy, don't worry about it because all of your cells are doing a whole lot of work like this. So we said, so this is sort of what we have understood and learned over the last 50 years. Most of this work, DNA was discovered, it was discovered earlier, but the structure was solved only in the 1950s. And over 50 years, we have understood how cells work to this level. Now, in the mid-'80s, there was a sense that if proteins are going to do everything, and all the proteins are encoded in the genes in your genome, then to really understand everything about how we work as humans, wouldn't it be good to sequence the entire human genome? And this was a radical idea. It might seem, today it might seem, why was there such a debate? But there was a huge debate in the biology community. Because some people said, oh, this is just big projects, big money. And it wasn't that easy to sequence DNA. But finally, the sort of people who wanted to go for it won the day, and the projects were funded. And in 2001, so it took 15 years to sequence the entire human genome. So we have 23 pairs of chromosomes. Each chromosome is made of DNA. And there are about 3 billion bases. And it cost about $3 billion to get this done. And then when you sequenced these 3 billion bases, you can analyze it and predict how many genes are there. How many genes does the human genome, taking all the DNA and all your 23 pairs of chromosomes, turned out there were only 25,000. And later, the genomes of the mouse were decoded, also of Drosophila, the fruit fly, and worms. And turned out there are actually the same number of genes in the mouse, the fly, and a worm. So, there's no reason for us to feel too great. They're about as complex. Well, that's not true. Actually, while the number of genes is the same, the sort of complexity comes in how proteins interact, in networks they make, in regulation of genes, etc. And that's the stuff we're just beginning to understand. So we now have the full genome sequence. And it's, while I said, it takes $3 billion to sequence the entire human genome, today it costs only about $10,000. So you can sequence a lot more. And this has really opened up the field. And it's something, that everybody knows that, there is huge genetic diversity in the human population. And even anecdotally, we know that, we often say, oh, people from the same family look alike, people from the same region look alike. And that's because genetically they are more identical. But on the whole, there is huge diversity. And that's because differences are present in the human genome between any two people. Of course, the exception is identical twins who get the same copy of the genome when they are born, and they certainly look very similar. So the next thing people started to ask the question is, how much diversity is there? And by diversity, what I mean is, if you look at, say, one region of some chromosome, if in one person there is a C paired with a G, in another person, in the same position, there may be a T paired with an A. And that is referred to as a single nucleotide polymorphism, or a SNP. And there's about one SNP for every 1,000 base pairs. So if you have 3 billion bases, there are between any two individuals. So any two individuals sitting in this room, there will be about 3 million differences like this. And they are what account for the differences between individuals. We have now identified about 7 million SNPs entirely, and there may be more, but it's about the range of 7 to 10 million SNPs that will be there in the entire human population. And now you actually can buy a small gene chip. And you can take DNA from a person, put it on the gene chip, and find out all the differences there are in between two individuals. Now, why is that important? Well, one of the things it tells us about human evolution. So between two people, if the genetic differences, so for example, people from the same region will have fewer genetic differences compared to people from very distant regions. And just by looking at how many genetic differences there are, because remember, the genetic differences or these polymorphisms, how do they appear? They appear entirely because of errors made during replication from generation to generation of DNA. So the longer time is, so it's like a molecular clock, the number of SNPs or differences there are. So if two people are from the same family or from the same region, so they have a same sort of founder parent, they will be more similar compared to somebody from two very different parts of the world. And based on that, looking at the number of differences, these SNPs between people, we can actually come up with a theory of human evolution, and the theory says that humans, Homo sapiens, probably evolved in Africa. And about 100,000 years ago, there were about 100,000 of them, and some of them, moved out of Africa and went to different parts of the world. And the most recent migrations were, so from Africa, some of them crossed over into the Middle East and went to Europe. Others crossed over and went to Asia and came to India. Others went further down south to Australia, etc. And some of them from Siberia crossed over the Bering Strait. This was, remember, happening at the time of the Ice Age, so that there was ice connecting these two continents, and they moved over into North America and then further down into South, and that was as recent as 10,000 years ago. Now, there are, as more data appears, there are small sort of differences to this theory. There's now some theory suggesting that when Homo sapiens moved out into Africa, there were already some Neanderthals already living there. And initially it was thought that there was never any crossing between Neanderthals and Homo sapiens, but the most recent data suggests that there may have been some crossing. So there are finer sort of tunings to but the overall theory still seems to be that humans originated in Africa, which is also why you have the most diversity, the most SNPs in African populations. And the least in the most recent migrations, if you look at South American, or, there's of course Native American or Native South American populations. So understanding how the human genome and the differences between genomes from different people tells you, and you can learn about how humans may have evolved and human history. But the other thing it does is it allows you to ask questions about human disease. Now again, I'm sure this is again in the popular, everybody knows that there are some diseases that are genetically inherited. And a simple example is if you take type 2 diabetes and ask the question, what is the risk if say for example, if there's somebody here in this audience who has type 2 diabetes, and if you ask, what's the chance that the person sitting next to him might have type 2 diabetes, if the person is unrelated, the risk is 5 to 10%. But if the person is sitting next to a sibling, then the risk is higher, it's 30%. And if you're sitting next to an identical twin, then the risk of the identical twin having type 2 diabetes is more than 80%. So what that tells you is clearly there is a genetic component to diseases such as type 2 diabetes. The question is, what are the genes that are responsible for type 2 diabetes? Now, why is it important to know what genes are responsible? Well, because the first thing is if you know what is the gene responsible, you'll then know what protein is responsible, and then you will know the root cause of diabetes. And then you can target that and think about ways to develop a drug against that protein that is misbehaving. So this is how drug development works. So the key is identifying the right target. Now, you may have heard, again, that it's very expensive making drugs. It costs about $800 million to come up with a new drug. Now, why is that? The high cost is really because you also have to pay for the failures. So when you develop a drug, it won't always work. And one of the key reasons why drugs fail is we don't know what is the real target we should be attacking. And what genetics allows you to do, as I'll show you in a minute, is have a much better grip on identifying and validating the key target, the root cause of the disease. And once you know the gene, you know the protein, you can study the protein, you can study what shape it has, what function it does, and then you can make a drug targeting that protein. And once you make a drug, you can then, if you have an animal model for the disease, then you can test it there and then go on to clinical trials and test it in humans. So that's how drug development works. So knowing the root cause, knowing the genes involved in diseases that have a genetic component is key to reducing cost of drug development and improving our chance of developing drugs. Now, many diseases are caused by single genes, so mutations in single genes. And these are simpler to deal with in terms of understanding them. So the gene is directly responsible for the disease. The environment has a very minor role. So for example, hemophilia or sickle cell anemia. So sickle cell anemia is caused by a mutation in the gene for hemoglobin. So we know the mutation. We know that another matter that we haven't really been able to come up with a cure for sickle cell anemia, but we know the cause. Hemophilia is a single point mutation in a clotting factor because of which your blood can't clot. So these are simpler, but these are actually not the rule. These are the exception. The most common diseases, such as diabetes, heart disease, hypertension, and many mental illnesses, schizophrenia, bipolar disorder, these are more complex. Genes only play part of the role. So genotype is important because we know some of these diseases sort of go with families. There are families in which, for example, schizophrenia is more common. There are groups of people where heart disease is more common. But it's not everybody with that mutation will end up with the disease. There's also a real role, an important role of the environment, of behavior. Smoking, for example. I say this sort of in an audience of architects and hope nobody throws things at me, including Bimal. And there's chance. For example, many viral infections. So infection with the Coxsackie virus in children often ends up, if you have the risk, sort of the risky gene, you might end up with juvenile diabetes. So there is also bad luck. So all these things put together, you end up with disease. And so this is more difficult to analyze, but now knowing the human genome, knowing all the genes, and knowing the SNPs, where genetic differences might be, this becomes a much more tractable problem. In the way you can ask the question now, in cases where genetics plays part of the role, but an important role, is you do what are called association studies. And the way the study is done is you take an affected population, so a population, say, with hypertension, and normal controls, people with normal blood pressure. And then you look at all the SNPs and all the genes, and ask the question, which gene or which polymorphism is found more often in the affected population? So for example, the triangles, if you count the numbers, is about the same. The blue circles is about the same. But these brown box representing a SNP on chromosome 12 at position this is more common here. So that might be involved in the risk to hypertension. And this has really opened up the field in the number of diseases for which we have now a clue about what the involved gene and the involved protein might be in the last 10 years is really impressive. So for example, just to give you one example, there's a disease called age-related macular degeneration. This happens in people usually about 60, 70, and what happens is in the retina, in the central part of the retina, in the macula, you get the growth of blood vessels. And because of that, you lose the central vision. So a person with AMD, if these two boys are standing in front of them, won't see the boys because this is where the image is formed in the macula, the center of the retina. And we had no clue what causes AMD. Are there any environmental factors? Are there any genetic factors? And using this kind of association study, we have now, so this genome-wide association study comparing SNPs between AMD patients and control groups, a single mutation in a protein called complement H was identified as the cause of disease. Now, complement H is actually a protein from your immune system. You would never have guessed that this will be involved in a disease related to the eye. But that's the data. And in addition, we know that there is one amino acid change. So in a normal person, at position 402 in that gene, instead of a tyrosine, you have a histidine. So tyrosine is one amino acid, is replaced by a histidine. And if you have that histidine in place of tyrosine, that increases your risk of AMD threefold. That's if you have one copy. Remember, we all have 2 copies of every gene. If, unluckily, you have both copies with the mutation, the risk is 9-fold. Now, that still doesn't help you very much today. I mean, if I'm told, my mother actually has AMD, so I'm actually curious what my risk is. But how does that help me? Because I don't know what to do to avoid AMD when I'm 70. Because we don't know what causes it. I mean, does more sunshine? If I have the risk of AMD, if I have this gene, if I face more sunshine, does my risk increase? I don't know. If it is, I'll wear goggles all the time. But I don't know yet. But it gives you a direction in trying to understand. And certainly, drug companies can try and understand how complement H leads to AMD and come up with treatments to target complement H. And that's the kind of work that is now progressing. The other thing genetic differences do is they change your response to different drugs. For example, to give you an example, there is a very effective drug called 6-MP, forget what the real chemical name is, which is very effective treatment for childhood leukemia. But 10% of the children have a toxic reaction. In fact, they can die from treatment with 6-MP. And nobody had a clue. How do I find out if this kid is going for this kid, if 6-MP is going to work or if it's going to be toxic? And they did a similar association study, and they found a polymorphism in the gene for one enzyme called TPMT. Again, don't worry about the name. This enzyme normally inactivates 6-MP. So 6-MP is very toxic. It does its job, but your system needs to take it out of the system quickly. And if you have this one single amino acid change, then the enzyme is there but not as active. And so in those children, this drug accumulates and it becomes toxic. So now there is a very simple test based on DNA where you can see if the child has this polymorphism that puts them at risk to 6-MP treatment. And if they are, then all you need to do is give them 10 times lower dose. And the drug still works and the children still recover. So this is how genetic diversity, understanding genetic diversity and the response to drugs will become sort of the future of medicine. You can imagine, some years from now, all of us, today the cost of sequencing the genome is $10,000. And it's predicted in another 5 years it might come down to less than $1,000, even $100. So if all of us, knew, and we knew which drugs, so drug companies will sort of come with a pamphlet saying, if you have this polymorphism, then you will need more of this drug or less of, so you can tune your response. And so that's the future of medicine. But that all sounds like it's all going to happen in the first world. What about the age-old problems such as malaria, death? So there, the rupture, these tiny green parasites, they're swimming around. One of them finds this red cell, it flips this red cell around, and finally goes inside. So it's a very fast process. You have a minute or two, less than a minute actually, to target this parasite. And so really need to understand how this parasite works. And this is an electron micrograph using electron microscopy looking at the parasite going in. So this is a parasite and this is the red cell. The parasite has a front end. So once it binds, its front end faces the red cell. Then you see this thickening of the red cell membrane. And then as the parasite moves in, you can see this is called the junction. It sort of now moves around. If you think in 3 dimensions, there is this junction all around. And it finally goes inside the red cell and will be enveloped by a membrane. So it's a complex process. And you would imagine there would be many molecular interactions between this parasite. It has to recognize this red cell. It has to make this junction and finally drive this process of invasion. So where do you begin? Well, you need to know what is the receptor on the red cell that the parasite binds to, and then what's the protein on the parasite that binds to this receptor. And we work both on Vivax and falciparum, but in the interest of time, I'm only going to talk about our work on Vivax. And this shows you sort of work going back to the 30s, where these people are in Africa. They're not on safari looking for lions and giraffes as we might do, but they're actually looking for malaria parasites. And such malaria safaris found that there's plenty of falciparum malaria, but no Vivax malaria in Africa. At the same time, in the 50s, not in the same time, about 20 years later, when blood transfusions and blood group typing sort of expanded around the globe, it was found that 95% of people in sub-Saharan Africa were Duffy negative. What does that mean? That means on their red cell, they're lacking a protein called the Duffy antigen or Duffy protein. And Lou Miller, who I worked with at NIH in the 70s, put together these two observations. And he wondered, this observation was there lying around for 20 years. Nobody bothered to think about it. And he wondered whether Vivax uses the Duffy antigen to bind to red cells and invade. And if you are Duffy negative, you won't get invaded, and maybe that's why there's no Vivax malaria in Africa. And it turned out to be right. So he tested Vivax, its ability to infect Duffy positive and Duffy negative red cells, and he showed that if you're Duffy negative, you won't get Vivax infection. Unfortunately, all of us here in India are Duffy positive. So don't bother to go around testing if you're Duffy negative and if you're lucky. Everybody in Asia, Southeast Asia, we're all Duffy positive. So we're all at risk of Vivax malaria. So that was a key breakthrough. And you see how long it takes to make some of these observations. And that's why I started working with Lou Miller in the, when was it? In the early 1990s. And at that time, the next step was identifying the parasite protein that binds to the Duffy antigen. And that's what we were able to do. We identified the parasite protein called the Duffy binding protein, which binds the Duffy antigen. And we know this must be critical for that invasion process. Now, this is a huge protein, so very difficult to make a vaccine with the whole thing. So the next important thing we did is by various methods, we identified what part of the protein is actually involved in binding. And it's this red part here, which we call region 2. And that's what we've been working on now for the last 10, 15 years. After I moved back to India, so a lot of our work has been on trying to understand how this molecule works. How does it bind to the Duffy antigen? Remember, proteins fold up, so they have to have the correct structure. And we actually, using X-ray crystallography, with colleagues at ICGEB, solved the crystal structure. And this is what the molecule looks like, only this bit. Because that's the bit that's important. That's the bit that's involved in binding. We then identified within this molecule, where are the amino acids that actually interact with the Duffy antigen? And this is the binding site, this bit in dark red and dark blue. So that's the part that actually binds to the red cell. And that's the part that you want to target. Now, an important thing, one of the reasons it's been difficult to make vaccines for parasitic diseases is, again, this is something you may have heard, is parasites change all the time. There are many different strains. So the question was, if we target this, will this look the same in all the Vivax strains? And turns out it is. This is highly conserved, which means every Vivax strain we have looked at, these amino acids look the same and the structure looks the same. So if we target this molecule, we should be able to target all Vivax strains. And so the next thing was to find a way to produce this protein in large quantities. And then the idea is, if I have that protein, if I immunize you with it, you'll make antibodies. When the parasite comes along, your antibodies will block the binding of the parasite with the red cell and protect you from infection. So we developed methods then over the last 10 years to produce this protein. And the way you do it is, because all organisms use the same genetic code, we can just take the gene from the malaria parasite, this bit that produces this binding domain, put it into a bacteria, and the bacteria will start producing that protein. And then we purify that protein, and this is the kind of lab, these are our fermenters, where we've put the gene from the bacteria, from the malaria parasite into E. coli, a bacteria, which we grow in this fermenter, and then the bacteria starts making the protein, we purify it, and we can make about 200,000 doses from just a liter of this bacteria. And now the next steps, so we've developed the process, we've shown that this molecule, if you immunize animals so far, they make antibodies, and the antibodies will kill the parasite. And in fact, the next step is we are going to bring our process to a company here in Ahmedabad called Intas Biopharmaceuticals to produce the vaccine for testing in human clinical trials. Now, using the same approach, we have also made a vaccine for falciparum malaria, and that is already in a clinical trial in Bangalore. Now, to end with, I'm going to tell you, so what we've done here for malaria is taken just a little piece of Plasmodium genome for this gene, for this one protein, and put it into a bacteria. And we could do that because all organisms use the same genetic code. But now, and you know how to write the genetic code, right? You know the sequence of A, T, C, G. If I have this, what protein I'll get. And we know a lot now about how cells work, how bacteria work. So what Craig Venter and his group at a company called Synthetic Genomics did, they took the bold step of actually writing DNA. So far we have been reading DNA. We read the human genome, we read the mouse genome, we read the bacterial genome, and to understand how these things work. But now if you understand how cells work, how microorganisms work, you can actually write DNA. You can write the sequences of genes and put them together. You can produce DNA, so there you can synthesize DNA. So there's a machine you can buy called a DNA synthesizer. You put 4 bottles of ATCG, you feed in your on the computer the sequence of the DNA, and the machine will give you DNA pieces with that sequence. So far we can make only about 1,000 base pair sequences, but then there are ways to stitch them together. And so Craig Venter's group sort of put together a synthetic genome with all the genes they thought would be required for a cell to survive, a microorganism, a single cell, a bacteria to survive. And they did another trick. They put in this one gene for antibiotic resistance to some antibiotic. And then they took this genome and stuck it into another bacteria and then allowed this bacteria to grow, and they added the antibiotic to the plate on which it was growing. So the original so when this bacteria gets this genome, the bacteria will grow, it will divide. Some of them will get this genome, which you have inserted. Others will get their own genome, the one they were born with. But those won't survive because they won't be resistant to the antibiotic. And so the ones that grew up were the ones with your synthetic genome. So you sort of wrote your DNA program on the computer. You used a machine to produce that DNA, stitch it up, stuck it into a bacteria, booted that cell. The cells grow, you select with the antibiotic, and you got bacteria growing. And this is a field called synthetic biology. So you are now actually writing DNA to create new organisms. So you design a minimal genome on the computer, synthesize the fragments, assemble the chromosome, transfer it to a bacteria, select for the new species, and what if these bacteria sort of get out in the environment? So you want to put some marks on the DNA, which are called as watermarks, to identify them, and I'll come to what they have put in the DNA. And these are the bacteria that they have produced, called Mycoplasma mycoides, because most of the genes they used were they learned from this organism called Mycoplasma, which is one of the smallest genomes, one of the simplest organisms. And they also put in a gene so that if you grow this on a particular medium, these microorganisms will turn blue. So you can identify them. So this is the first species where the genome was designed on a computer, where the genome was synthesized in the laboratory on DNA synthesizers, and to create this new species. And now what is the application of this? Well, the application is, there are many applications. So one, there are all kinds of microorganisms, for example, in the ocean. So Craig Venter actually sailed across in the Pacific from Japan to the US or something and collected buckets of ocean water. And he went back to the lab and saw what microorganisms they are. He studied them, and he found all kinds of organisms. So there are organisms that will take carbon dioxide from the atmosphere and turn them into methane. And others that will produce all kinds of other things, that will produce ethanol. Now, if you understand the genes involved in taking carbon dioxide and producing methane, you can imagine what you can do. You can solve your carbon dioxide problem, and you'll solve your energy problem. So that's the kind of thing that people want to do. You can think of pathways and stitch together genes in an organism to produce ethanol. So you're no longer dependent on Middle East oil. So there are all kinds of applications in creating new organisms that produce things that you can use. And I was going to come back to watermarks. So to identify these bacteria that they've created, first of all, in the DNA, they put in a code. Now remember, ATCG, 4 bases, can be used to make a code to represent 20 amino acids. So ATG is for methionine, TCA is for something else. So you can do the same thing. You can write the English alphabet, where all you need is 26. So the code is in the genome of this bacteria. And what it spells out, if you get this bacteria and sequence it, it has the names of Craig Venter and his co-workers. It gives you a website where you can go to and read all about this bacteria. And there are 3 quotes. And the quotes are, to live, to err, to fall, to triumph, to create life out of life. And this is a quote from James Joyce, from the Portrait of an Artist as a Young Man, where he wrote a biography of Vincent van Gogh. And he was probably talking about life out of life, meaning creating a painting that looks like life, but these scientists have sort of taken it a little literally. And 2 physicists. And I'm glad I did physics, because after all this, they had to quote 2 physicists. What I cannot create, I cannot understand. Richard Feynman, this is actually something he just wrote on a blackboard in Caltech, which has become famous. And see things not as they are, but as they might be, by Robert Oppenheimer, who was involved in the Manhattan Project for the first nuclear bomb. And so this is, to me, this sort of tells you the thinking and the ethos of scientists working in this field. They want to do sort of the next big thing, solve problems, understand life, and think of ways that one could not have thought of before to solve some of the problems that we are faced with. Now, I would like to warn you, there are, in all the things I've described, this is sort of opening up huge new potentials for addressing problems of health, or addressing problems of the environment, or addressing problems of energy. But one must remember that scientists are often, we would like to believe and think of the potential of sort of positive outcomes, of problems we can solve. But we must always be aware that there can also be undesirable consequences and things that we may not think through early on. And that's something that you as society need to think about. That's why you need to understand what's going on in science so that you can create opinions and direct, what out of all this, what should we use and what should we just let go by. Thank you very much. Evolution. So HIV, the sort of most accepted theory for HIV is there was a similar disease in monkeys called simian immunodeficiency virus. And in Africa, people have actually a very close relation with monkeys. They're often pets, they often eat monkeys, they eat brains of monkeys, it's considered a delicacy. So somewhere, it must have jumped the species barrier and come to humans. And so the way it is tracked, and again, you can use genetic differences to sort of, did I kill your camera? So you can track genetic differences in HIV strains and go back to where it might have originated. And it's thought it may have originated in Central Africa, from there gone to the West Indies, and from there to the United States, and from there to the rest of the world. So that, to me, as a scientist, is the most reasonable theory. There are all kinds of other theories. Americans introduced it, which I don't quite buy. This, to me, is the most reasonable theory. Yeah. Change DNA of microorganisms, we do all the time. If you're asking of Okay, for humans. So there was the big hope. In the 1980s and 90s, there was the big hope, you must have heard of gene therapy. That if I have a genetic disease, then I just replace that faulty copy with a copy that works. And gene therapy was very popular and funded hugely. But unfortunately, that just didn't work. It worked in the test tube. It never worked even in animals. So there were some very famous gene therapy experiments done. There was, if you remember, at the NIH, there was this little boy called the bubble boy, because he was born with a mutation in one of the immune response genes, because of which he was susceptible to any little infection. I mean, he could not handle any infection. So they created a bubble which was sterilized, and he lived for, 10, 12 years in that bubble. Food was sterilized and passed on to him. So his interaction was entirely through the bubble, in the hope that they will one day find a way to solve that problem. And this was actually while I was at the NIH. So they found the gene, they found ways to replace that gene in the test tube, and what they did was basically take all his blood out, replace the gene, and put it back into this little boy. And he survived some, but the experiment just didn't work. So there are many such examples. So gene therapy has really not turned out to be the promise that it was thought to be. And in fact, gene therapy funding for research, unless there is some really new breakthrough to solve the problem is one of efficiency. How efficiently can you replace, you have so many cells. How can you replace the gene in all the cells? Till you can make some technical breakthroughs, it's still not likely. Yeah.
How do you actually connect genes and intelligence? As evolution was taking place, genes were kind of doing things to replicate itself and self-select or naturally select to grow. And then we have this whole human intelligence thing. Would it be something like genes would fight back? Because right now we are, say, getting away from the natural selection process? Or where does the genes and the chemical things connect with the intelligence of replication?
So things like intelligence, there's not going to be a simple answer, because it's not going to be one gene for intelligence. I find it, and so it's going to be things like intelligence are, first of all, very difficult to even quantitate. People don't agree on which IQ test is the best. There's culture dependence, etc. So we don't even know how to measure intelligence. And it's unlikely that there'll be a single gene for intelligence. It's complex of genes. That's where environment, actually, maybe your early schooling plays a big role in how you think. People are now discovering that actually the first few months of life, being stimulated by various things, influences how you think later. So this is a very difficult question. I don't think it's going to be a very simple answer to things like, what genes influence intelligence. Yeah. So that's the same thing. Unless you're looking for a trait, and unless you can quantify that trait, it's very difficult to address the question. So there are some questions you can't address in biology.
Last question. Okay. Yeah.
You showed us those processes in RNA is made, and they looked very animated, looked like machines, basically. But we know that they're molecules and chemicals, right? And they're not alive. Is
not
there
actually
alive, right? Is there a stage of complexity which you call, before which you don't call it life, and after which you call it life?
Yeah, so these are individual processes. I'm not suggesting that, just that specific process we zoomed in on is life. It's one of the processes that you need to learn. So, and this is a difficult question. How do you define something? How do you define living from dead? So typical things you look at are, one is there metabolism? So it's taking nutrients and producing energy. Can it replicate? Can it divide? And so for that, you need a whole lot of processes happening. All these things, even if you mess up DNA replication, you'll kill. Even if you mess up protein synthesis. So for example, protein synthesis, I mentioned all our cells do protein synthesis, all organisms do protein, but there are minor differences between the ribosome in a bacteria and a human ribosome. And once you understand the differences, that's how you actually design drugs that will target protein synthesis in a bacteria and not affect a human cell. So life is sort of all these processes happening together, and some of the key features to distinguish living from dead is metabolism and the ability to replicate.
I think, okay, last, and then maybe we can carry the conversation after the lecture.
Can you throw some light on cloning, what they have been able to do on that ship, and how cloning they are able to do?
So cloning of whole organisms, you basically, you take the DNA or the genome from one sheep, whose characteristics you like, you take an egg and remove the DNA, and so you take this one which has both properties, and you then basically fuse it, you insert it into the egg, and then add some chemicals to trigger the process of division. And if you do all this right, you basically initiate, you trigger, you sort of start off the process of division in a way that would happen naturally. And the first part of it you actually do in a petri dish. And once it comes up to a certain stage, you can't have a sheep embryo growing in a petri. So at the right stage, you take it and implant it into a female sheep, that's whose hormones you have, properly adjusted and injected to make its, sort of placenta and uterus, everything ready to accept an implant. And then the mother sheep takes over.
You can carry on the conversation. I understand it's a provocative topic. Thanks, Dr. Chitnis. Thanks a lot for coming here. Can I have a round of applause for Dr. Chitnis, please? It was wonderful because you really unpacked extremely complex scientific concepts in a simplified lecture form for us. On behalf of the Faculty of Architecture, FAAA, and everyone here, a small token of appreciation from Apurva. Thank you. Frankly, I didn't know there's so much action in our body and so much hard work. I understand now when our students say that they've been working hard. So I'll take that with a pinch of salt. Okay, there's a small, since I have the mic, small announcement to be made. It's not an announcement, something important. It is really about FAAA. For the last 1 year, a lot of you are aware of it, but some of you are new faces here from other postgraduate program. For the last 1 year, we have been witnessing quite a unique and an exciting phenomena in the city or all across the country, where we find a new institutional structure coming up in the form of FAAA. That's the Faculty of Architecture Alumni Association. It is exciting, it is unique because it really tries to harness the positive energy of alumni all across the world into something meaningful and a formal engagement. It is very exciting for us because as I see it and as our colleagues and students see it, it offers possibilities for relationship and collaboration at a different level from an institutional perspective. We also realize that to make FAAA a success, it would need our engagement, our institutional engagement with FAAA would be very, very critical. And we wish FAAA all the best for its endeavor, and we will be cooperating in all the things that we do together. Thank you. Thank you so much. And the next lecture is after 2 weeks, September 24th. Please be here. Thank you.